Welcome to video 3.1. In this video, we'll be starting to talk about protein structures by introducing the first fundamental building block of protein, the amino acid. So, much like DNA and RNA, proteins are polymers, and they're polymers of amino acids. Let's quickly break down what an amino acid is structurally in this box, and then we'll move to talk more about it. And of course, the polymers they make, proteins. So, amino acids start. The acid part is a carboxylic acid. So I'll draw that on this side, where we have a carbon connected to a double bond to oxygen and an OH on the same carbon. That is your carboxylic acid. It's connected to a central carbon, and we call this the alpha carbon. And then there is an amine, so the amino part is this nitrogen, NH2. So we have the amino, we have the acid, we have an amino acid. Now, what makes amino acids different is what we put on the alpha carbon, right? The fundamental thing is, as long as you have something connected here, you have an amino acid. And that's really the core structure of the amino acid. So compared to sugars, it's quite simple, right? Looking down to the key functional groups, again, we have the carboxylic acid. And important things to note about the carboxylic acid is that they can take on a negative charge. Actually, at pH 7, which is the most relevant pH for biology, right, carboxylic acids are always negatively charged. It's only once you get to a pH of around 4, pretty acidic, that you start seeing the regular form. So if we go back up here, we actually put a negative charge on this oxygen. Amines are similar, right? Amines, NH2, are actually going to be positive most of the time in biological systems. So we're going to see this being NH3+. So this is called the Zwitter ion, when you have both of these charges correctly drawn at pH 7 of the amino acid, just named after some guy named Zwitter, probably. But again, these are your key functional groups. There's overall no charge. There's a negative and a positive. There's a carboxylic acid in the negative form and an amine in the positive form with something connected to the alpha carbon. That is all there is behind an amino acid. Now, why is this important? Right? Imagine a polymer of 50 different residues. With DNA, we have 4 acids to choose from. We have G, C, A, and T. So 50 different polymers, right, are 50 different things. We could have 50 Gs or 50 Cs. There would be a lot of different combinations. But again, every time you're picking the next letter, you only have 4 choices, right? G, C, A, or T. With amino acids, there are 20 different amino acids that are encoded for, right? 20 different acids means that if you have 50 polymers, right, there's going to be so many more combinations. And if I ask you how many unique combinations are there for 50 residues, well, ignoring some complicating factors, right, there's 20 amino acids to the power of 50. That is a very, very large number, which is to say that the total space, the total number of combinations to make protein are just so much higher than there are for DNA, which is why we see all the different structural diversity in the family of proteins. More amino acids means more different combinations. Some terminology for the polymers of amino acids. If we have less than 40 to 50, and this is a round, not the best number, it's not like there's a firm number where this name changes, but generally small sequences of amino acids are called peptides. Peptides. Where larger amino acid proteins are called polypeptides. Right? Poly just meaning that there's lots of them. Right? Again, all of these, it's just a small thing versus a long thing. It's not like there's multiple peptides within a polypeptide. But at the end of the day here, we can say that it's a big, long protein. We use the term polypeptide. It makes it what is a protein? It's when a polypeptide has function. So we name, we don't just have names for every combination of a protein, but we give proteins names when we realize that they have some sort of function. Another thing to consider when you're naming proteins, right? So this is a long string of amino acids, but here it's kind of shown all balled up and you'll see why that makes more sense in a little bit. But there are proteins that are monomers, so a single chain, a single polypeptide, one polypeptide, will fold and form a protein. But there are also different types. There are dimers, trimers, tetramers, right? Proteins that are actually made up of multiple polypeptides, right? So overall, and this is the interesting thing, we would call this whole thing a protein, even though it's made of two distinct different chemicals, right? We have polypeptide 1 and polypeptide 2, and they come together to form the dimer. Two different polypeptides come together. Dimer means two molecule, and we form the protein. So proteins are just functional units of amino acids, and we can talk about their lengths this way: peptides, polypeptides. So hopefully that helps paint the stage a little bit for what's to come. Of course, we're going to dive in more to the spaghetti. But before we go any further into the amino acids themselves, let's talk about the bonds that they make. And the important idea here is the term peptide bond. So two amino acids here are depicted. We have amino acid 1 and amino acid 2. Both of them are going to form a peptide bond. So I'll write that down here. And it's very similar to what we saw with DNA. Right? If we look at this amino acid, again, we see its amine group, we see its carboxylic acid. Its carboxylic acid is going to be able to react with the amine group of another amino acid. What's going to happen is we're going to keep the nitrogen. Again, without getting into the pure chemistry of it, we'll get rid of the OH, and we'll take one of these H's. So those are going to leave, again, as water. It's a dehydration reaction, just like it was with DNA as well. And what we're going to see now is the nitrogen. So this is amino acid 1 with its R group 1. So whatever's connected there tells you what the amino acid is. We'll talk about that more later. There's the carboxylic acid, but instead of being connected to its oxygen, which is left as water, it's now connected to the red nitrogen of carbon 2, or rather of amino acid 2. That nitrogen is still connected to its carbon and then R2 and a carboxylic acid. And here we've made a tumor or a dipeptide. Right? This is not to be confused with a cancer tumor. It's a dimer or tumor. We'll call it that sometimes. Now, here we see that the peptide bond is the thing connecting the two amino acids. The peptide bond is an amide bond. So it's a carbon that's double bonded to an oxygen and connected to a nitrogen. That is what a peptide bond is, and it's fundamentally the thing connecting amino acids to make proteins, which make up you and I. There are a couple key features of a peptide bond that are worth keeping in mind. So here I've shown a threemer, or a tripeptide. We can see that there are two distinct peptide bonds. The first connects the black and red molecule. The second connects the red and blue molecule. And I've drawn these as squares because there's an interesting feature. Remember these lone pairs? Beside amides, we're able to do resonance. Again, we're not going to have to draw these forms out in biochemistry, but this feature, the fact that amides have resonance, means that this part of the molecule is actually quite rigid. It's not able to rotate as freely, right? This part of the molecule can totally rotate freely, but the peptide bonds can't. What that leads to is something that's quite interesting. I like to view it as a spaghetti versus skipping a rope that has those annoying plastic tassels on them if you've ever used them, right? A protein is not like this. It's not just pure spaghetti that folds. It's little pieces, so this part, the alpha carbons, right, in between the different amino acids, they're able to rotate. But the peptide bonds themselves, because of the resonance they have and their stability, which we'll talk about, are unable to rotate. And so you really have this like kinked chain where the rigidity of these things really affects the folding. And that's why we get a lot of predictable folding out of proteins, as you'll see. Again, another thing to mention is that these are very stable, right? Amides, without going into the chemistry too much, are one of the more stable functional groups that exist. And it's important that if we're made of protein, our protein just doesn't randomly break on us. So with that in mind, that is some features of the peptide bond. Now, jumping to directionality, Recall that in DNA and RNA, we had a direction. There was a 5' free phosphate and a 3' free hydroxyl group. With protein, we have something very similar. Right? I'll pull the sequence down below. You can see that on the left side here, we have a free N. We call this the N terminus. So the side of the amino, or the side of the protein that has a free amine, right? The other amines are all trapped up in the peptide bonds inside. But the free amine, that's going to be your start. That's the first amino acid when you read the direction of the protein. We go R1, R2, R3, we're going through the amino acids, and eventually we get to a free carboxylic acid. That would be the C terminus, right? A free acid. And this is a free amine, right? So we treat this as the end of our sequence. So we read proteins from the N terminus to the C terminus. That's the direction that we read it. Now, let's play a quick game of spotting the difference. So let me just quickly erase this. Pause the video once I've erased these X's, and try to spot the difference between these two chemicals. And see if you can identify, are these the same? Are they different? And if they are different, how? Great. So looking at these, we can see there are a couple key differences. The first is that while it does look like the R groups are in the same place, these are not the same sequence. The reason for that is that the N terminus on the first molecule is here, so this is the start. So we would read this protein sequence as R1, R2, R3, R4, C terminus. This is 1 to 4. On the bottom here, though, even though it's drawn the same way, the free N terminus is not on this side. We're not always going to be reading left to right. The N terminus is here. So we're actually going to read this 4, 3, 2, 1, and this is the end C terminus. So this is 4 to 1. So these are actually different peptides. These are different proteins despite looking very similar and being made of the same amino acids. Directionality is important. Lastly, let's just do one quick overall comparison between DNA and protein before moving on and digging into the amino acids themselves. One thing to keep in mind between DNA and protein is that they're both similar in that they are built of blocks. For DNA in particular, we have the nucleotide, and for protein, we have the amino acid. Right? When we look at how these things are connected, in DNA, we connect them by phosphodiester bonds. Right? I'll even write that down, phosphodiester. And here is nucleotide. This is an amino acid. And it's linked by a peptide bond. Right? An amide bond. And instead of having these bases to choose from, we instead have different R groups, different things connected to our amino acids. There are 20 of them instead of 4. So lots more flexibility. The other last key important difference is that if we look at the backbone of DNA, right, and when we say backbone, I mean everything that isn't the R groups or the Cs, the things here that are in black, right? I see there's lots of negative charge. Every phosphate brings along with it negative charges. And these can be quite unstable, right? It's important to recognize that those charges are there and need to be stabilized. With the amino acid, though, the backbone itself has no charge. Right? The R groups might, who knows what they are, but there's no charge on the backbone. There is, however, a lot of hydrogen bonding. We'll talk about that in the next video, and hopefully now you can see how the similarities between making polymers for DNA and for protein. With that, I'll see you in the next video.